Fabian Leon joined our program almost 2 years ago. He's finishing up a master's degree, and all goes well, he'll be transitioning into a PhD program with us. Fabian came to us from Kentucky. He grew up in Kentucky. His family is originally from Mexico, and I think some of his interest in the agricultural sector comes from his family's background in farming operation in Michoacan. And he joined our program after finishing up a degree in ag biotechnology there at the University of Kentucky, and has jumped into some of the work we're doing around genomic prediction and looking at new ways to use technology in a sorghum breeding program. Sorghum is, as you guys know, a lot like corn in its breeding processes, but doesn't have quite the private industry support in that area that corn gets. And so application of some of those technologies is interesting, and it's something that we're starting to pursue right now. And I think he's going to start to introduce that concept today. And so with that, I'll let Fabian take it away.
Thank you, Dr. Rooney. Are you all seeing the slides?
We're seeing it and we're hearing you good.
Okay, is it full screened all right, Dr. Finlayson?
It is.
Okay. Thanks again for the introduction, Dr. Rooney. Today I'll be presenting about the evaluation and prediction of the performance of hybrids derived from the elite lines relevant to our program. And some of the lines that I'll be talking about are actually in this group photo that we took. The block that we're standing inside of are the parental lines I'll be talking about. And then over my shoulder, you'll see a section of the hybrids that we're going to be testing. So for this presentation, I've sort of planned this kind of outline. I do first want to talk about an introduction to sorghum breeding and give people the context to understand what my project is. And then I'll talk about what my objectives are and some of the results I found. And also, I will also talk about the future work that I'm going to get to. Because Dr. Rooney, like Dr. Rooney said, I do have plans to kind of stay here and keep working in this space. First and foremost, sorghum bicolor is the species that I'm working with. This is what our breeding program is all about. Sorghum's a self-pollinated diploid that does have some outcrossing. It is a C4 grass. It's got the high photosynthetic efficiency of a C4 grass, and also of importance is its small genome size. It's about 25% of the size of maize or sugarcane, and it's fully sequenced, which is great for my research and the applications of what I do. ICRISAT says that sorghum is grown in 103 million acres worldwide, so it's definitely got a big footprint and big large-scale impacts. And its success and its reach is kind of due to some of its drought tolerance, resilience to poor soil conditions, its diversity in morphology, and also its diversity in the end uses that we use sorghum for. So a lot of the world eats sorghum. A lot of it uses it for food, fuel, fiber, and feed. And in the United States, we're actually the leading producer of grain sorghum. And according to the USDA's National Ag Statistics Service, we planted 5 million acres of sorghum last year here in the States, and Texas accounted for 1.8 million of those. This map on the right here depicts the sorghum acreage by county, and that's a great display of the sorghum's reach throughout Texas. As you can see, a lot of those green counties are in the Panhandle, and grain sorghum has really established itself successfully in rotations there in the Panhandle, in Central and in South Texas. So in the United States, sorghum is actually grown as a hybrid crop, and hybrid seed production for sorghum is enabled by the ABR cytoplasmic male sterility system, which delineates the male and female heterotic groups for sorghum. And A lines are actually at the top here. A lines have the nuclear and cytoplasmic genetics for sterility. Those do not shed pollen, and thus those are used for, they're used as the female in hybrid seed production. The B lines here are maintainer lines. They're nearly identical to the A lines, but have normal cytoplasm, and therefore they can be crossed with the A lines to maintain stocks of the A line. And finally, the restorer lines here shed pollen, have nuclear genetics for fertility restoration as well. And that's actually what allows the hybrid progeny to shed pollen and ultimately set seed in the farmer's field when the farmer grows it. So here in our program, we have historically been concerned with breeding and releasing parental ABR lines. And we've kind of let the private industry concern themselves with making the hybrid combinations and releasing elite hybrids. Since A lines and R lines are the products that we deliver, we have to have some metric for choosing which parents are better than the rest. And because the parental line's purpose is to yield strong hybrids, we evaluate it by using it in hybrid combinations and phenotyping its progeny, and then cycling back to attribute those measures of combining ability back to the parental line depending on how its progeny performed. So the average or overall performance of a line across hybrid combinations is dubbed its general combining ability, and the performance of a line in a specific combination is its specific combining ability. This is exactly where my project comes into play. In my experiment, I have been given a set of 48 elite R lines and 46 elite A lines that are highly relevant to our breeding program. A lot of them are from our program, some are from other public breeding programs around the states. This dendrogram on the right was generated using the SNP genotyping data from my experiment, and it sort of depicts the relatedness amongst my lines. You can see two distinct heterotic groups here with the A lines in red and the R lines in blue. And you can even see where some of the female lines from outside programs like Kansas State group together here. So the objective for my program, or for my project specifically, is to discover the best hybrid combinations possible from these parental lines and sort of measure the genetic merit of each one. The challenges of doing this is that there's 48 males and 46 females, and that would mean making and evaluating 2,208 combinations of hybrid combinations. And this is just not really feasible for our program's resources, time, money, and effort. And without making every single possible hybrid, we also not seeing a complete picture of a parental line's genetic merit. So for my project, the desired outcomes are to generate and evaluate the hybrid combinations that we can make, and also use those to develop a genomic prediction model for the rest that we can't make. So to carry out my project, I did follow this step-by-step timeline that I'll be talking about. I want to first present steps 1 through 3, generating the hybrids, evaluating them, and analyzing the data to attribute combining abilities to the parental lines. So I'll dive into that first. In the summer of 2018, the program set out to make as many of these hybrid combinations as possible with these parents. And they succeeded in crossing 399 unique combinations or dots, as you can see up here. But the point of this figure is to kind of display just how much missing information we have, or just how incomplete this factorial is. So all that white space is actually what we're trying to fill in. The 399 hybrids that we did make, we actually split in half into 2 sets. Actually, 1 hybrid was used in both sets. So we had 2 sets of 200 hybrids, and these sets became the 2 trials that were planted across the state. In 2019, both sets were grown in College Station, and the first set was also grown in Taft, Texas. In 2020, both sets were grown again in College Station. And this year, the second set was sent down south to Monte Alto, Texas. The trials were grown in a randomized complete block design with 2 replications, and standard agronomic practices for grain sorghum production were used. The traits that we were interested in phenotyping here were for yield, height, days to metanthesis, and panicle exertion. Days to metanthesis is actually flowering time, so we measure that when half the plot has flowered halfway down. So the data that we actually collected from those yield trials had these distributions for yield on the left and plant height on the right. The data seem to be distributed normally for the most part. And with these violin plots, we're also able to visualize the differences between College Station and South Texas environments. The latter, which had lower yields on average, it looked like. To answer the question of which hybrids in the entire experiment were the best, I conducted a linear mixed model analysis with that data, and I tried to explain yield with hybrid environments, reps within environments, and hybrid by environment interactions. And I considered the year location combinations as my environments. So I have 4 unique environments. And for the model, hybrids and hybrid by environment terms were considered random effects, while environment and rep within environment were fixed effects. The best linear unbiased predictions, or like the calculated coefficients for the random hybrid effects, were used to rank the hybrids. So that's the distribution of those BLEPs there on the right. For yield BLEPs, we were obviously interested in individuals with higher or more positive BLEP values. So from this distribution, we're actually more interested in those shown up to the right side of that distribution. I'm also showing you the variance explained by each source of variation from an all random analysis, just to depict what's affecting the yields we're seeing, and just how much each source of variation accounts for. I do have the same linear model to explain plant height and calculate BLEPs for the hybrids plant heights. In this figure on the right, we were actually interested in the individuals on the lower end of the distribution, just because shorter plants are actually what the sorghum industry prefers. They actually like shorter grain hybrids. And I do have the same variance explained model here on the left. So to attribute what we're seeing in the hybrids back to the merit of the parental lines, I actually used a similar linear model. And in this model, I broke up the hybrid term from before and replaced it with terms representing the male, female, and male by female interaction that was used for the hybrid. And these terms are supposed to represent or reflect the general combining ability of the males, the females, and then the specific combining ability of their combinations. And like before, I'm showing you the variance explained from the terms in this model. And in this analysis, I also drive BLEPs for each effect. And you can see these distributions for yield BLEP amongst the R lines and the A lines here on the left. Here we're interested in working with the lines on the far right of these distributions, since those are now said to have higher general combining ability. Lines with higher general combining ability can and will be used with our breeding program going forward, just to test cross with and kind of compare future progeny with. So that does have that practical importance for our program. And those are the takeaways from the first steps in the methods. And now I want to talk about the latter steps of my project that actually include the genotyping and the genomic selection that I've started to wade into. The genotyping for the inbred lines was actually primarily carried out by Dr. Klein and Dr. Klein and their group in the Borlaug Center. They followed a genotyping by sequencing protocol outlined in this Morshigi et al. Paper with the NGO-MIV restriction enzyme, and produced a SNP marker matrix. And the resulting SNP marker matrix was further processed and handled by my fellow sorghum breeding student, Jalis Fonseca, who actually used the markers to build a genomic relationship matrix for the parental lines. In that genomic relationship matrix, I actually took and subset male and females to build a genomic relationship matrix for my hybrids. And that's what I actually used for genomic selection, or from my genomic prediction model. So just briefly, in case you're new to genomic selection, I do want to explain the approach and why we built the prediction models and equations the way that we did. So to build a model that predicts if a hybrid is going to yield really well or really poorly, you first have to train the model with a population for which you have both genotypes and phenotypes. And from this training population, you can determine the relationships between genotypic markers of each parent and the resulting phenotypes of the hybrids. And once that model is trained, it should be validated on a separate breeding population to see how well it does. In my project, actually, the two sets of hybrids that I described actually serve as delineations between a training set and a validation set. And the model that I used is actually called a two-step GBLE model, or can be described as a two-step GBLE model. In the first step, I actually calculate means for each hybrid across the test. And in the second step, I actually augment my BLUP calculations for hybrids with the genomic relationship matrix of the hybrids that I mentioned in the last slide. The results of that and the prediction accuracies are shown here for this GBLEP approach. The accuracies are Pearson's correlation coefficients between the predicted and observed values of the hybrids, and the prediction accuracies on the left are the result of training the model with my first hybrid set and then predicting the second, and the accuracies on the right side column are the results of training the model with the second set of hybrids and then predicting the first. So this figure on the right does show the scatter plot sort of fit of predicted versus observed values for yield when training the model with the first set of hybrids and then predicting the second. And while I do have that model and those results, I still have a lot to learn in terms of genomic selection. So I do have a lot more future work to do here with this project. And going forward, I do want to study more, learn more about the G/E analysis and the stability of these hybrids. I also need to try to do a more robust validation scheme for my GBLEP model that might allow for it to be trained with a larger portion of the hybrids that I've used, and then maybe validate it iteratively, kind of use some of that k-fold cross-validation for that. And lastly, there's a lot of other genomic selection approaches that I have yet to learn and implement. So there's a Bayesian models and obviously the deep learning models that I haven't looked into or tried, but that our program is working on and has some preliminary results for. But that's the work of other students. So a lot of the sorghum breeding students right now are kind of exploring this space and trying to see what we can develop. So with that, I will thank the Sorghumville community and all the other students and everybody who's kind of enabled me to look at this project. So thank you to Dr. Rooney, Dr. Klein, and Dr. Klein for serving on my committee and for kind of directing me through this process. I also want to thank Dr. Hoffman, Dr. Hodnett, Delroy, Vicky, and Steve, and previous students and current students, Dr. Horn, Dr. Harvey, Amani, Jollis, Nick, Daniel Mitchell, Noah, and Kayla. And then from the Klein's group, Dr. Patil, Julie, and Lauren, who have all been out in the nursery with me at some point or helped me take notes at any point too. And lastly, I do want to thank the organizations that are kind of backing my master's education here, shown on the bottom. So thank you. And are there any questions?
Nice job, Fabian. Very nice job. Questions?
I have a question, Scott. Amir Ibrahim is here. Very good talk. I wonder if set 1 and set 2 are the evaluation and prediction sets, right? Am I correct?
Yes, training and validation sets.
And these were not drawn at all locations, because in two locations you only had one or the other set.
Yeah, in South Texas we only set one at a time.
And so when you showed that R-square, or actually correlation between predicted and actual of less than 0.3 for yield, which is not very high actually. So seems like you cannot predict yield at this point. You can predict height, but yield is the goal ultimately. These values, are these the overall values, or because you might need to look at the within location, because at colony station you have both sets, right? So did you try to look at within location prediction accuracy?
Yeah, we haven't done a within location prediction accuracy. One of the ways that I tried to approach what you're talking about was with the two-step GBLE. I first did attempt to kind of arrive at those means across environments and the means for each hybrid across environments explained by that linear model, and that's actually what I fit my model to. So it should be across all the observations for that hybrid in the trials.
And the hybrid is fixed, right? It's a fixed variable in your model.
For the actual GBLE prediction, yes.
Yes, yeah, because that's how you get actually those least squares means.
Mm-hmm. Yeah.
Okay. Well, how would you improve that accuracy in the future? What are your thoughts for improving it?
Yeah, hopefully some of the cross-validation will tell me more specifically just how good the model is, or be better indicators of how good the model is. And then also what you're talking about, we can also kind of incorporate environmental variables into the models, or maybe just look within an environment. But the end goal would hopefully—
any covariances that you can actually think about, include in the model? Do you need to include any covariance? Because you have differences across locations. When you have one predictive set in one area, and then you have an evaluation set in another area, you might have variables. So that's one area you can look into, I guess.
Yeah, definitely.
Thank you so much.
I had a question. I was just curious more because I know that your program also looks at sorghum varieties high in nutrient composition, like in phenolic acids and tannins. Just curious if you thought this approach might apply to those traits as well, realizing I think that typically those have a high proportion of non-additive variants. Just curious your thoughts, really.
Yeah, that's definitely something that we'll have to try, and I know that some of the other students are already thinking about those questions. I don't see why we couldn't try it and get some sort of accuracy. I don't know that at the end of the day that's going to be useful for breeding for those traits, but we do a lot of NIRing and measuring those. So if those are phenotypic values that we can fit a model to, can't promise a great model that's going to be useful, but it's definitely might be worth one of our students' exploration.
Thanks.
Well, I'll ask, so you showed a nice genomic prediction experiment. What do you think the strategy, or what would you recommend for moving into genomic selection and actually beginning to base things off of this? I mean, are the marker sets going to stay the same, or are those going to change? How many more years of data do you think you need till you, if you were running the breeding program, till you would feel comfortable in making decisions based on this?
So actually, I think, can you repeat the first part of the question?
Well, I mean, this was, you didn't actually base any selection decisions off of this, so it was predictions. Do you feel like it worked well enough that you would make those selections based off of this, or do you think you need some more years of data? And then are those marker sets going to be consistent?
Yeah, so going forward, the marker sets are actually going to hopefully stack on top of each other. So what we actually did was build a genomic relationship matrix, which is actually dependent on what marker set you have. So if you add more lines, you have to go back and recalculate all of that again. So in that sense, we need to update the model every year. In another sense, I was predicting hybrid combinations. So there's not really anything predictive or genomic selection about what hybrid combinations to make. I mean, I can recommend that we make certain crosses this summer because those are going to be good hybrids. But I haven't actually investigated what lines are predicted, what lines kind of have higher general combining ability beyond just what we've observed in those trials. So I know Dr. Rooney is already looking at some of that data just to know which lines to test cross with going forward in the program. And because we have those lines already genotyped, they will flow into the genomic prediction approach that we're going to be using and rolling with going forward. About how confident I feel making choices with my predictive accuracies, not very confident. I mean, I can tell you what some of the bad, maybe what some of the bad hybrids are going to be, but I cannot very confidently right now say or predict what good hybrids are and which ones we haven't made that we need to make.
Hey, but not making the bad ones again is worth something, right?
Yeah, it does save time and effort if you don't waste your time on those. Yeah.
Other questions? There's a question in the chat for you, Fabian.
All right. Were the Pearson's correlation coefficients between predicted and observed values significant? I did not test for significance, so I can't tell you. I'd have to look back at my data. I'm sure I got the output, but I didn't present it on my slides. So,
well, significance means that the correlation is not equal to 0 in the original population, so it doesn't make any difference. As long as these are low values, they are low values.
Yeah, and that's the takeaway to present.
Other questions? Going once, going twice, going 3 times. Thanks for a great talk.